All right, everybody, welcome to Chapter 2.1 on Molecules to Metabolism. Okay, so this is one of those chapters that you should really pay close attention to because the things that we're going to talk about in this chapter are going to carry forward through every single topic that we cover for the rest of this course. Okay, so let's start off with listing the basic types of biological macromolecules. So those are carbohydrates, lipids, proteins, and nucleic acids. These are the four types of large organic molecules that we're going to use as the basis for all the structures in living things. Okay, now I alluded to those molecules as organic molecules, so let's take a moment to figure out what exactly that means. Okay, organic molecules contain carbon, so that's going to be an important thought here, and most of them contain carbon-carbon bonds. Okay, so we have to be a little bit careful here because there are some molecules, like my friend carbon dioxide over here, that contain carbon, but they're not organic, okay? So this guy is not organic, and that's because it doesn't have any carbon-carbon bonds, right? So we have carbon double bonded to oxygen over here. I can draw those in. There aren't any bonds between carbon atoms, and so that's a little bit different than this molecule. Okay, here's an example of an organic molecule that is going to be your best friend for the rest of this course, and this is, of course, our friend glucose. Okay, so I would get prepared to recognize, draw, dream about this guy. Okay, this is glucose, and you can see here that glucose contains lots of bonds between carbon atoms, and I'm not going to draw them all in, okay, but we can see that there are bonds between carbons, and that's one of the things that makes this an organic molecule. Okay, now let's take a trip in our time machine back to our study in chemistry and talk about carbon. So carbon has 4 valence electrons, and that means that it's down for some bonding, okay, that it's going to be able to make 4 bonds in order to fill that outer shell, which can hold 8. So let's recap. It has 4, that outer shell can hold 8. That means that it's likely to make 4 bonds. And the way that it's going to do that is by making covalent bonds. So covalent bonding, you may remember, involves sharing electrons. It does not form ionic bonds. Ionic bonding involves gaining or losing an electron. It forms 4 stable covalent bonds. Okay, so I should always be thinking of carbon having 4 bonds, and that can be like this case where this carbon has 1, 2, 3, 4 single bonds. It could be 1 single bond and 1 triple bond. It could be 1, 2 single bonds and 1 double bond. It doesn't matter. You just need to know that it forms a total of 4 bonds. Those bonds are extremely stable, okay, and those are covalent bonds. And that's really the property that makes it so versatile and the basis for all of our other biological macromolecules, okay? So it's able to form those wonderful stable covalent bonds, 4 of them, with either itself, so we can see some carbon-carbon bonding sometimes, or it can form those bonds with other types of elements. So it can do it with oxygen, hydrogen, sulfur, phosphorus, nitrogen, a whole host of other elements, okay? And that's what makes it the backbone of these important macromolecules. Okay, now I keep saying the word macromolecule. This is one of those wonderful, amazing times where this word is exactly what it sounds like. Macro meaning big, and then molecule. It's a big molecule. Well, we have another term for that, and that term is polymer. So mer means to make, okay? Poly means many, to make something out of many small parts. And those many small parts are called monomers, okay? So mono meaning one. I can take a bunch of monomers and put them together to make a polymer. So you may hear them called, these polymers, called macromolecules, polymers. You may just hear them referred to as a monomer, as a molecule. In terms of these monomers, you may hear them called monomers, you may hear them called subcomponents, you may hear them be referred to as building blocks. Doesn't matter, it's all the same. Okay, so we've got our 4 basic macromolecules here. Carbohydrates can be turned into polymers by using monomers of monosaccharides, like single sugars hooked together. Lipids, we can build large lipid molecules by attaching fatty acids to a molecule called glycerol. Proteins are an example of a polymer, a long strand of molecules. We also sometimes hear them referred to as polypeptides, although really these words mean different things. We'll find more about that later. Just for now, we need to know that they're associated with each other. And those are polymers made out of small building blocks called amino acids. And then we have our nucleic acids, right? So things like DNA. And that is a long strand of molecules called nucleotides. Okay, so here's where life gets a little bit complicated, right? So I just told you that carbohydrates can be built out of monosaccharides, simple sugars, but they don't have to. They can stay as simple sugars. So I'm going to give you a minute just to kind of like copy this down. Carbohydrates can be broken down into 3 different groups, okay? Monosaccharides, disaccharides, and polysaccharides. You need to be able to name examples of each of these, okay? So you're going to have to categorize these as monosaccharides. You may also be forced to just name them. You may get a prompt that says, state 3 monosaccharides. Okay, well, that's a little scary. Same thing we hear with disaccharides, maltose, lactose, sucrose, polysaccharides, starch, glycogen, cellulose. It is okay if you can't remember these or you don't know what these are right off the bat. We're going to do a whole chapter on carbohydrates, a whole chapter on proteins, and so on and so forth, okay? So for now, let's just get these down. Let's commit some of them to memory here. And then later on, you'll be glad that you did this, because once you know more and then you're using these notes to prepare for the IB exam, all of these words are going to make sense to you. So now's a good time to pause the video and jot those down. Okay, so in addition to naming and recognizing molecules, you also know how to recognize pictures and, yes, how to draw them. So you don't need to draw them right away because we're going to do single chapters on this, but some molecules you may want to know are glucose, fatty acids, particularly saturated fatty acids, ribose, and amino acids. But again, we'll have more on that stuff later. Okay, so when we talk about your metabolism, what we're really talking about is a sum of all the chemical reactions. So what is a chemical reaction? Well, a chemical reaction is some kind of a collision between molecules that results in a change in those molecules. Okay, so here we're watching a collision between 2 molecules. There's some sort of a collision, and then afterwards, our 2 molecules look very different than they did in the beginning. Okay, most molecular collisions don't result in anything. It's just one thing bouncing off of another, and they look exactly the same. Some collisions, however, result in enough energy to actually have a chemical reaction, to actually change. So what determines whether or not those collisions are going to result in a chemical reaction? Well, there's a couple of factors. Okay, it depends on the type of molecules that are colliding. Okay, if they don't have some kind of compatible shapes or anything else, then we're not going to see a chemical reaction. They're just going to bounce off of each other and go on about their merry way. The second thing is the orientation. So that's kind of like the positioning of the molecules when they collide. And the third is the speed of the collision. Okay, speed is kind of one of those measures of how much energy those molecules have, and that speed can often determine whether or not a chemical reaction takes place. So in thinking about temperature, if I increase the temperature of an environment, what that does is it increases the speed. And the more speed they have, the more likely they are to have a collision that results in a chemical reaction. Okay, so oftentimes we have to raise the temperature to get things to happen. Now, that's going to have some pretty terrible effects if we raise the temperature too much, but more on that later. Okay, so since we can't rely on being able to raise the temperature enough to make molecular collisions result in chemical reactions, okay, cells often use proteins called enzymes in order to increase the likelihood that those reactions will occur. Now, we're going to do a whole topic on enzymes later, okay, but for now you need to know that they are proteins and that they help catalyze biochemical reactions. So they make it more likely that a chemical reaction will take place once these molecules collide. And they do that by lowering something called activation energy. Activation energy is the energy that you have to put into a reaction to get it started. Okay, and so what enzymes do is they help put molecules in the correct orientation to make it more likely that their collision will result in a reaction. And this also results in them needing less energy when they collide in order for a reaction to take place. So we say that it lowers their activation energy. So here's kind of what an activation energy graph looks like. You have to put energy into a reaction to get it going, to make sure that those molecular collisions result in a reaction. Enzymes orient molecules in such a way and weaken their bonds in such a way that you have to put less energy into that collision or less activation energy in order for that reaction to occur. Okay, so here's a great example of how this works in your actual cells, a real life example. For this example, we're going to look at a molecule called ATP. ATP stands for adenosine triphosphate. Okay, so it looks something like this. Adenosine, which is a combination of adenine and ribose, adenosine, and then triphosphate, 1, 2, 3 phosphate groups. Now, this is a high energy molecule. When cells need energy, what they do is they break the chemical bond between the second and third phosphate groups. Okay, and when we break that bond, all of the chemical energy in that bond gets released for the cells to use. But then we no longer have ATP. We have ADP, adenosine diphosphate, so adenosine with 2 phosphates, and then this broken off phosphate group. Eventually what's going to happen is you're going to eat something and your cells are going to undergo cellular respiration, and they're going to use some of that energy from your food to reestablish this bond, to put these two back together. Well, the odds of this and this just randomly running into each other with enough force to actually recreate that bond are very low. So what your cells do is they use an enzyme. They bring this phosphate group and ADP together and orient them in such a way that they're able to rejoin, okay, that reaction is able to take place, and it happens a lot faster. Okay, now when we say metabolism, I know sometimes we think about heart rate or respiration rate or how quickly we burn calories, but that's kind of a gross misinterpretation of this word. Okay, when we say metabolism, we're talking about the sum of all of the biochemical reactions in a cell. And here they are. Okay, if you take biochemistry in college, this is what you're studying. Okay, this is a roadmap to all of the chemical reactions that would be taking place in your cells. We're going to cover a few of them, like the replication of DNA, synthesizing RNA, synthesizing protein, cellular respiration, photosynthesis. We're not going to go through all of them, okay? But when we say metabolism, we mean a sum of all of these chemical reactions going on in your cells. It's also worth noting that almost all of these are catalyzed by enzymes. Okay, so all of these crazy looking reactions require these guys are enzymes. Okay, now all those crazy reactions can be put into one of two categories. We have catabolism and anabolism, or anabolism, however you want to say it. I have to be honest with you, I never remember both of these. I only remember this one, okay? And here's how I remember this. I'm a child of the 90s, and in the 90s, all the professional athletes, it seemed, were getting busted for using what we call anabolic steroids. So anabolic steroids helped them to build muscle, okay, get bigger, faster, etc. Okay, so I remember anabolic as something that builds. Okay, so what does that have to do with your metabolism? Great, well, let's take a closer look at that. Okay, so anabolism is building large molecules by putting together many small molecules. So here's small molecules. Anabolism builds them into larger molecules. Now, this involves those condensation reactions, okay? So we're putting things together by removing water. We'll talk more about this later. This requires energy, okay? Putting things together requires energy because we are building or making bonds. And when you create bonds, that requires energy. Okay, so all I do then is remember that catabolism is the opposite. If anabolism is building, catabolism is breaking things down. Okay, so another way to remember this is catabolism sounds a little bit like catastrophe, so things falling apart. Up to you how you want to remember it. But breaking down large molecules into smaller. So something like this, and breaking it down. These use hydrolysis reactions, okay, and that literally means to break using water. Lysis means break, hydro means water. These release energy, and that's because we are breaking bonds, okay? So when you break bonds, you release the chemical energy held in those bonds, and so catabolism releasing energy. So a couple of examples here, okay, involved with, I'm thinking particularly digestion. When you eat food, you break down the large food molecules. That's catabolism. So here's food, my digestive system breaks them down. When these small molecules get absorbed into our bloodstream and then carried by our blood to our cells use these small molecules to make larger molecules of their own, and that's called anabolism. Okay, so let's talk about these hydrolysis and condensation reactions for just a minute. Hydrolysis reactions involve splitting molecules by adding in water. So again, these are associated with catabolism, splitting, catabolism. Again, that's going to release energy, and that process requires or consumes water. You're going to know you're going to have to know how to draw these. Sorry for your life. Okay? So let's think about this. If I have 6 molecules of sucrose, okay, that are put together, so I'm not going to draw their actual shapes or anything. 1, 2, 3, 4, 5, 6. In order to break these apart, I'm going to need to add 1, 2, 3, 4, 5 molecules of water, because I need to add a molecule of water everywhere that I'm planning on breaking a bond, okay? And that's because it looks something like this. Okay, when we are breaking a bond, we're going to reestablish what our original molecules look like using hydrogens. So there's two hydrogens here, one of them goes here, another one goes here, okay, and an oxygen, and that goes here. And now you can see we've reestablished what glucose and fructose are supposed to look like. Okay, we've broken this bond here. Okay, here's another example of hydrolysis, this time involving an amino acid. Okay, amino acids are held together by something called a peptide bond, more on that later. But again, okay, in order to break this bond in here, I need to add water. One of these hydrogens is going to go here, the other hydrogen is going to go here, and the oxygen is going to go here. And so to really understand this, you have to understand how amino acids are put together. Notice now that their structure is identical. So we've turned this into these two by adding water. Okay, reestablishing the original structure of the amino acids. So next up, we're going to talk about condensation reactions. These are the opposite of hydrolysis reactions. So here, we're going to be joining two molecules together by removing water. Let's take a look at these two amino acids. They're identical. In order to join them together, we're going to remove H2O, so an H from one and OH from another, that makes H2O or water. And then this carbon and this nitrogen can form a bond together. Now, since we're making bonds, this is going to require energy. Notice that reactions that require energy release water. In hydrolysis reactions, they require water, but they release energy. So they kind of work on the opposite scale there. All right, you're going to have to know how to draw these eventually. But let's talk about this question down at the bottom. This one should say how many water molecules are released. Okay, so how many are released? Wow, that says released. That is awful. Anyways, to connect 5 amino acids. So let's say I have 1, 2, 3, 4, 5 amino acids, and I want to connect them. Well, this is going to release a molecule of water every time I want to join one together, because joining one removes a molecule of water. And that means in order to form these bonds, I would need 1, 2, 3, 4 molecules of water. So every time we're making a bond there. Okay, so here's a look at the condensation reaction of 2 sugars. Okay, these are monosaccharides, 2 sugars. And I know that condensation, okay, removes water. Okay, in order to form bonds. So where is that water going to come from? Well, it's going to come from here, these 2 OH groups. Okay, so where is this water molecule coming? Because there's only 2 H's and 1 O. Well, I'm going to remove these 2 H's, okay? And I'm going to remove this oxygen also. And then this carbon and this carbon can share this oxygen. Okay, and that's going to fulfill all their bonding requirements. Oxygen has to make 2 bonds. Carbon has to make 4 bonds. Okay, so if I remove this OH group from this carbon, it's only going to have 1, 2, 3 bonds. It needs a 4th bond. If I remove this hydrogen, then this oxygen is only making 1 bond. It needs a 2nd bond. So what's going to happen is this oxygen is going to bond to that carbon. And in the end, if you can get through kind of like all of my mess here, in the end, it's going to look like this. Okay? It's also important that you add this to your reactions. So while we're on the subject of molecules, okay, let's talk about kind of how we came to an understanding scientifically about how these molecules work. See, way back in the day, there was this thing called vitalism. Okay, and vitalism was the long-held belief that living things were kind of playing by different rules than non-living things. And I can understand how people thought that, right? Because living things were made up of organic molecules and non-living things were made up of very different types of molecules. Okay? Now, what they were finding over and over and over again was that organic molecules could only be synthesized or made by living things. So let's take this plant here. This plant is taking inorganic molecules like carbon dioxide and through some kind of mysterious magic, turning them into organic molecules like sugar. And it was thought that because of that weird and strange and wonderful conversion, that only living things like plants were subject to these laws and were able to do this. Creating organic molecules was thought to be something that only living things could do. That's called vitalism. Well, one of the organic molecules that was pretty easy to study at the time was something called urea. Okay, so urea looks like this. Okay, it's an organic molecule produced by lots of living things. And in mammals, urea is excreted in urine. Okay, so urea was used by vitalism scientists a lot. Because first of all, they could get a lot of it. That was pretty easy to harvest. And they did studies on it and showed that only living things could produce urea along with other organic molecules. So the use and study of urea was kind of confirming this concept that only living things could produce organic molecules. And then along came this sexy guy. Okay, this is Friedrich Wöhler. And what Friedrich Wöhler did is he mixed two inorganic chemicals together. And well, he did this a lot of times unsuccessfully, but finally he was able to get these inorganic chemicals to react with one another to form urea. So basically what he did is in a laboratory setting, outside of a living thing, outside of the cells of living organisms, he was able to turn something inorganic into something organic. And it was the first time that it was shown that living things weren't the only things possible or able to create organic molecules. So the idea of vitalism kind of began to crumble, and this discovery led to many more types of organic molecules being synthesized in laboratory settings. And this was super important in terms of kind of figuring out how life on Earth first originated. Okay, how did these organic molecules like proteins and DNA come to be on Earth? Well, they had to start out in some kind of non-living, inorganic space and then move forward from there. And so this guy had a super important breakthrough in terms of that discovery. And that'll do it for chapter 2.1.